Hemostasis is a Greek word meaning stoppage of bleeding. It is the process that arrests bleeding at the site of injury and maintains blood in a fluid state within the closed vascular system. Hemostasis involves major and minor systems. Major systems include platelets, the vascular system, and the coagulation system. Minor systems include the kinin system, fibrinolytic system, serine protease inhibitors, and the complement system. Hemostasis is further divided into primary and secondary hemostasis. Primary hemostasis involves platelets and blood vessels. Secondary hemostasis involves coagulation factors, coagulation inhibitors, and the fibrinolytic system. Hemostasis occurs in 4 phases: vascular spasm, platelet plug formation, blood coagulation factor activation for clot formation, and activation of control mechanisms, including inhibitors and the fibrinolytic system. Platelets are structurally divided into 3 zones. The surface membranous structure has a phospholipid bilayer and glycoproteins essential for normal functioning, such as GPIA, IC, and IIBIA. The second zone is the cytoskeletal zone. This system supports the platelet membrane and open canalicular system. Microtubules help maintain the discoid shape of platelets and are important for their formation. The third zone is the granular zone, which contains alpha granules, dense granules, lysosomes, and peroxisomes. The first function of platelets is their role in primary hemostasis. The second function is platelet procoagulant activity. Vascular injury activates the coagulation cascade. Platelet surfaces protect activated coagulation factors from inactivation, and they release fibrinogen and antiheparin factors. Platelets also maintain vascular integrity by repairing and nourishing damaged endothelium. Platelet-derived growth factor, or PDGF, stimulates vascular smooth muscle cells, hastening vascular healing. The coagulation system involves plasma proteins that play a major role in secondary hemostasis. Definitive hemostasis is achieved when fibrin is added to the platelet mass, followed by clot retraction. This slide provides additional details on the coagulation system. Note that factor 6 is no longer used. Other coagulation factors include Fletcher factor, also known as prekallikrein, and Fitzgerald factor, or high molecular weight kininogen. Together, these factors coagulate blood through a classical pathway. The classical pathway is divided into 3 parts: the intrinsic pathway, the extrinsic pathway, and the common pathway. The intrinsic pathway components are present in the bloodstream and include FXII, FXI, FIX, FVIII, HMWK, and prekallikrein. It is activated by contact with a foreign surface, such as a damaged vessel wall. The extrinsic pathway requires a factor not normally present in the blood, known as tissue factor, or FIII, which is released upon cell injury. FIII then binds to FVIIA and calcium. The common pathway involves 3 steps: activation of FX, conversion of prothrombin to thrombin, and polymerization of fibrin. Factors involved are FX, FV, FII, FI, and FXIII. This diagram illustrates the coagulation cascade.